Good afternoon, everyone, and welcome to the Recover Research Review, or R3 seminar. My name is Claire Quiner, and I'm an epidemiologist with the Recover Administrative Coordinating Center and the moderator of today's seminar. The goal of today's seminar series is to catalyze a shared understanding of the research within the Recover Consortium. And I want to start by thanking everyone who submitted questions in advance. Please submit any questions that arise during presentation, the Q&A feature in Zoom. After the presentation, we will answer as many questions as possible. A Q&A document will be posted with the recording of the seminar on recovercovid.org. It will include the answers for submitted questions today relevant to today's presentations. Questions about other scientific topics will be addressed in future seminars, and answers to broader questions about Recover will be available in the FAQs at recovercovid.org. As a reminder, we cannot answer individual questions about clinical care. Next slide, please. Our presenter, another couple of slides. There we go, our panelists. Our panelists today are Dr. Catherine Blish, Dr. Cliff Rosen, and Dr. Avindra Nath. And our discussant today is Dr. Sudha Seshadri. Dr. Catherine Blish is a George E. and Lucy Becker Professor of Medicine at the Stanford University School of Medicine. She has a joint appointment in the Stanford Immunology Program, is the Associate Dean for Basic and Translational Research, the co-director of the Stanford Medical Scientist Training Program, and is an investigator of the Chan Zuckerberg BioHub. Her research is dedicated to learning how to harness the immune system and to prevent immune responses into a diverse array of pathogens, including HIV, dengue virus, influenza, tuberculosis, and SARS-CoV-2. Dr. Clifford Rosen is an endocrinologist and bone biologist. He currently runs an 11-person basic and translational research laboratory at Maine Medical Center Research Institute. His focus is on understanding the relationship between bone cells and fat cells in the marrow niche and the role of parathyroid hormone in defining lineage allocation. Dr. Nath is the clinical director of the National Institute of Neurological Disorders and Stroke at NIH, where he is also chief of the section of infections of the nervous system and director of the Translational Center for Neurological Sciences. He specializes in neuroimmunology and neurovirology. His research is focused on studying the clinical manifestations and pathophysiology and developing treatments for neurological infections with a focus on HIV infections and endogenous retroviruses. Dr. Sudha Seshadri is a board-certified neurologist and the Robert R. Barker Distinguished University Professor of Neurology, Psychiatry, and Cellular and Integrative Physiology at the University of Texas Health Sciences Center in San Antonio, and serves as the founding director of the Glenn Biggs Institute for Alzheimer's and Neurodegenerative Diseases. She leads a neurology working group, several of them within several large consortia such as CHARGE and Recover, and is PI for other large national and international consortial studies on genetics and vascular brain injury. Her research interests are in using clinical, epidemiological, genetic, and multi-omic approaches to identify the biology underlying healthy brain aging, stroke, VCID, and ADRD, and to study possible preventive and therapeutic interventions. The topic of today's seminar is slightly updated from when the meeting invite was first sent out. It's the impacts of SARS-CoV-2 infection on brain, immunity, and metabolism. Today's speakers will share our current understanding, the gaps in our knowledge, and how Recover will contribute to filling these knowledge gaps. With that, I'd like to hand it over to Dr. Catherine Blish.
Okay, and I assume I've got the right view going. I think I do. Not seeing disagreement, so it must be okay. Just moving a few things so I can see. So it's really a pleasure to have the chance to talk to this large group about some of the work that my lab has done looking at metabolic and immunologic basis of long COVID. And I'm going to start by very, very briefly mentioning some work that we've done and our interest in SARS-CoV-2 infection of adipose tissue. And I'm going to mention this briefly because this is an ongoing study for which, unfortunately, we're still at the enrollment stage and don't have a ton of new data. And the initial findings were actually published in this manuscript in Science Translational Medicine in late 2022, in which we found that SARS-CoV-2 can drive an inflammatory response in human adipose tissue by infecting both adipocytes and macrophages. And in this study, we were able to look at autopsy samples from about 8 individuals, and we found evidence of SARS-CoV-2 infection in 8 of them within the adipose tissue. But we also did some in vitro work looking at SARS-CoV-2 infection in a dish. And we found that we could identify infection both within mature adipocytes that was productive, as well as macrophages, which was an abortive infection. But this combination drove a broad inflammatory response, which induced the secretion of multiple cytokines that are associated with symptoms of long COVID. And this really led us to speculate that SARS-CoV-2 infection in adipose tissue could contribute to severe COVID-19 in obese individuals via 2 mechanisms. One is the potential of an expanded viral reservoir, and the second is inflammation in periorgan fat depots. And this is something that we've really been interested in following up on, but in particular, we're interested in whether this has any link to the post-COVID condition or long COVID. And of course, there isn't, unfortunately, a set single case definition of long COVID, which I'll allude to again later. But for the sake of this talk, I'm going to focus on largely the WHO definition, which is the development of symptoms or the persistence of symptoms 3 months after initial SARS-CoV-2 infection, with symptoms lasting for at least 2 months and no other explanation. And of course, we all know we're here because there's a massive burden of disease from long COVID. So, of course, the other reason we're all here is that we really want to understand what the mechanisms driving long COVID are so that we can develop new therapeutics and treatments. And there have been numerous really nice reviews on this topic. I'm just grabbing one picture from this nice review by Chen et al., in which the 3 primary mechanisms that have been discussed are the idea of viral persistence, persistence of immune dysregulation, or direct tissue damage that's been persistent. And of course, when it comes to our adipose tissue, we're particularly interested in whether this viral persistence could be related to adipose tissue infection and whether adipose tissue could be a reservoir for ongoing replication or inflammation, as has been seen in some other infectious diseases, including, for instance, HIV-1. And to this end, we're currently evaluating adipose tissue biopsies from long COVID patients to look for virus and inflammation, but unfortunately, we're not quite at a point where we have the data all back yet from sequencing to discuss. So I'm going to move on and talk about the second big area, which is that we're interested in, which is immune dysregulation and how that might contribute to long COVID. So again, same figure. This is the middle part. To some level of activation of the immune response or exhaustion, is that contributing to these long COVID symptoms? And the work from our lab that I'm going to discuss has really been led by an infectious disease fellow in my group, Rebecca Hamlin, who generated all this data. And Sean Pincos, a very talented pulmonary critical care fellow, has really pitched in on much of the analysis of this as well. And I want to begin with a very brief overview of what happens to the immune response during acute COVID-19. And of course, many groups, mine, many others, evaluated this. This was our first foray into this, which was the single cell atlas of the peripheral immune response, which we published early in the pandemic and made the data available in approximately May of 2020. And we followed with this multi-omic profiling. And really, there's been great consensus as to what the changes are in the setting of acute COVID in terms of peripheral immune dysregulation. And for instance, what we found is that monocytes and conventional dendritic cells take on a tolerogenic or immunosuppressive phenotype. We see hyperactivation of both neutrophils and NK cells, but ironically, this hyperactivation is associated with dysfunction as well. And we see changes in hematopoiesis, leading to the development of immature neutrophils and a reduction in the number of lymphocytes. And of course, there are many, many other studies that have shown similar or slightly different or expanded upon results. But in general, this has been a very clear picture. And what's less clear is, do any of these changes persist or change? And is that contributing to long COVID symptoms? And to that extent, I want to talk briefly about some of the data that has come out from other groups and place this in context for what I'm going to show. And of course, and I think Dr. Rosen's going to mention this as well, one of the most exciting findings recently has been the report that long COVID might be associated with diminished cortisol levels. And in the study by Klein et al. From Akiko Iwasaki's lab, which we heard about, I think, last month, it was the most significant predictor of long COVID in their cohort. Interestingly enough, this is one of the few signatures that's been seen in a completely separate study by another group in the Sue et al. Study led by Jim Heath's group and others. They also saw it decreased, but importantly, it was a little more nuanced in this study. It was only decreased in those with respiratory symptoms or with 4 or more symptoms, and those with more mild disease, it was not decreased. So maybe not quite a universal signature, but more work needs to be done. Another recent study that's been very exciting is the idea that diminished serotonin levels could be contributing to long COVID symptoms. In this nice study by Wong et al., in which they found in a large UPenn cohort, it decreased in long COVID patients compared to recovered patients. Now, this is important because comparing long COVID to recovered patients implies that this is associated with long COVID and is not merely something that is associated with viral resolution in general. Now, they evaluated 2 validation cohorts in this study. In the first, an Irish cohort, they also found decreased serotonin levels, but importantly, they did not have a comparator group of recovered COVID patients. This was compared to healthy controls. So it's not quite an apples to apples comparison. And in their third validation cohort, samples taken from the uncovered cohort from 2 to 24 months post-infection, there was no significant difference in long COVID in serotonin levels. And the authors speculate that different modes of patient recruitment, the number of symptoms, disease severity, any of these things could have contributed to these disparate results. But this really raises many of the challenges that have plagued some of these immune studies of long COVID, which is there are differences in how we're defining long COVID, in the timing of the samples, in the populations in which we're studying them. And these have all made it very difficult to find a broad, generalizable signature that we can really base a therapy on. So with that in mind, I love the immune system, and I want to know, are there changes in specific cell types or their function that are contributing to long COVID? And of course, we are only one of 20 or so labs who are evaluating this. And I thought it might be useful because Rebecca Hamlin, my fellow, has spent some time looking in detail at what else has been published to summarize some of the studies that have come out first before we talk about what we saw in our study and place that in context. And so what I'm going to show is kind of a painfully complicated table and long table, looking first at the innate immune cells in long COVID versus recovered patients. And just as an example, I've narrowed this down to one cell type, classical monocytes. And what I'm showing is several of the studies that have looked at long COVID, most of which are published, one of which is still in the preprint era. And if the box is colored gray, that means this cell type was looked at and there was no change between long COVID and a recovered patient. If there's a line through it, it means they didn't look at the cell type, so ignore that data. If it's red, that means this cell population was increased in long COVID with sometimes some notes, maybe it was in a subset of long COVID. If it's blue, it was decreased. And right away, you can see what my challenge has been in trying to find a unified signature. Right? So classical monocytes are the same in 3 studies in different settings. In one setting, depending on whether it's pulmonary long COVID, there's a different subset that's increased and one subset that's decreased. In another study, they're decreased. So that's classical monocytes. If we look at other cell types in which we've been able to find more than one study that have looked, what we see is a bit of a smattering. There's not a consistent innate signature that we've been able to identify that has held across multiple studies in multiple populations. Okay, so maybe the adaptive immune response is the answer. Here are some of the major adaptive immune cell types that have been evaluated. And what we're seeing is kind of the same thing. If you look at naive B cells, they're decreased in this study, the same in this study, and increased in this study. So it's very hard to come up with a unified signature. I'm not going to go through all the different cell types. But of course, this is just phenotyping in terms of gross populations. What about their function? And there have been several studies that have looked in particular at T cell responses to stimulation with either p-aminomycin, which is a generalized stimulation, or with SARS-CoV-2 peptides. And again, once again, what we see is really a challenge in finding a consistent result. So for instance, if we just look at these first two, both use p-aminomycin as a stimulation. And one found an increase in function, one found no difference. And then if we look at this Peluso study, again, a similar thing, a different stimulation though, a decrease in function, which then conflicts with this study. So with this setting, what we found we were initially surprised and confused by, but maybe it's just that different cohorts are different. And it's going to be challenging and we're going to need greater numbers to come up with answers. So with this in mind, I'm now going to step back and talk about some very preliminary analyses. And keep in mind, this is all very early analyses of a study that we developed early in the pandemic that we termed the Infection Recovery in SARS-CoV-2 or IRIS study. And this was a study that was built in collaboration with many of my colleagues in infectious diseases, Phil Grant, Aruna Subramanian, Hector Bonilla, Upi Singh, Pras Jagannathan, and Bonnie Maldonado. And we designed this study in June of 2020, began enrollment in August, before we even knew that long COVID was actually a thing, just to study the process of recovery from COVID-19 infection. And so we enrolled 100 patients in August of 2020, between August and December, with the idea of following them for 12 months post-diagnosis with biospecimens collected at both 3 months and 12 months and clinical data at 6 months post-diagnosis. So this is the battery of tests that we collected on these individuals. We had medical history and symptoms, a number of scores of anxiety, fatigue, work activity, neurocognitive testing, but importantly for my lab, serum, plasma, PBMCs, and PAX gene samples. So we had these biospecimens at these time points. And then we also, because many of these subjects were enrolled in our early outpatient trials of interventions for COVID-19, including the PEG interferon lambda and the favipiravir study, neither of which showed any efficacy, by the way. So we ended up grouping the placebos and the controls. We had some acute samples available. So with this cohort, we wanted to delve into, are there any immunologic changes in those with persistent versus not persistent symptoms? And so what I'm showing here is just our classic demographic table. And I'm sorry, I know it's kind of small. There's a lot going on here. We had, again, 100 total enrolled patients, 20 of whom were fully asymptomatic, and 80 of whom actually had some degree of persistent symptoms at that first 3-month time point. If we look, they're relatively well matched between the asymptomatic and persistence for age and gender, which is interesting because gender has actually been enriched and symptomatic in many other cohorts. We had a large percentage of Hispanic or Latino population given the demographics in our area. The BMIs were relatively well matched, and a fairly low frequency of these subjects were hospitalized or in the intensive care unit or intubated, and somewhat enriched in our long COVID patients in this. So what were the symptoms in this cohort? And they were pretty variable. So first of all, I already mentioned about 20% were asymptomatic. And by far the most common symptoms across all time points, and arguably even increasing when it comes to brain fog or memory impairment, were fatigue and brain fog and memory impairment. These were followed by symptoms including myalgias and dyspnea, headache, chest pain or tightness, arthralgias, and sleep disturbance. And then finally, we did see altered taste and smell, cough, and hair loss. And these were the only symptoms that consistently declined over the 12-month follow-up. So with this framework in mind, we felt like we could address 3 major research questions. The first is, what features of acute COVID predict recovery versus long COVID using those acute samples we have? The second is, are there immunologic differences at 3 months post-infection in those who recover versus those with long COVID? And finally, what immune features correlate with persistent long COVID versus recovery at 12 months? And this was our experimental study design. We used pre-pandemic controls as well. And we compared our acute COVID samples, for which we had 38, our 3-month post-infection samples, for which we had 100, and our 12 months post-infection. And Rebecca generated a really nice, massive data set by stimulating these PBMCs with a generalized stimulation cocktail to do a site-off assessment of the function of multiple immune cell subsets. We froze some samples for single-cell RNA sequencing and processed that data to look at the transcriptome. And then we used the plasma for a proteomic study using the Olink platform. Now, because these analyses are in the early days, and we don't have a ton of time, I'm going to focus just on some of the parsed single-cell RNA sequencing data that we did. And for reasons of cost, this is extraordinarily expensive. We chose the 96 samples for this analysis, those with the most symptomatic versus the fully recovered patients. And I'm going to focus just on one of our 3 questions in my remaining 5 minutes, which are, what are the immunologic differences at 3 months post-infection in those who recover versus long COVID? Okay, so what does this data look like at 3 months? First, here is a UMAP projection of our single-cell RNA sequencing data. So each of these dots on this UMAP represents an individual cell. We've downsampled this because if I showed every cell that we looked at, it would be way too many in a giant blob. But what you can see is that we can see all the major immune cell subsets, including some hematopoietic stem cells, as well as rare T cell types like gamma deltas and our major cell types. Oops. So to look at this data, one of the first things we did was we wanted to know what the transcriptional profile was of these individual cell types. And we used these blood transcriptome modules, which were described by my colleague Bali Palendran in this nice 2014 paper, to describe the overall transcriptional network analysis of known genes that go together. And the advantage of these BTMs over some of the KEGG or other pathway analyses you might know is that these were selected for blood transcriptional modules and are really focused broadly on immune function, more so than on cancer, which is how many of the other ones were made. Importantly, these BTMs were named for the cell type in which they were most prevalent, but that doesn't mean that these transcriptional pathways can't be expressed in other cell types, which will become apparent why I'm bothering to say that in a moment. So what did we see when we looked at 3 months post-diagnosis in our long COVID patients in red compared to our recovered patients in blue? And if we look first at this particular blood transcriptional module, we found that this was downregulated myeloid and dendritic cell activation via NF-kappa B, one of the canonical transcription factors for immune cell activation, was downregulated in 3 distinct cell types: CD14 monocytes, conventional dendritic cell type 2s, and CD16 monocytes. So if we then looked at additional transcriptome modules, we saw a transcription factor also associated with activation and a B cell transcription factor also downregulated in CD14 monocytes and CDC2s. And finally, multiple pro-inflammatory DC and myeloid cell responses that were downregulated in our classical CD14 monocytes. So we didn't see much outside this myeloid cell compartment, but we did see a downregulation of an inflammatory module in natural killer cells as well. In terms of modules that were upregulated or more pro-inflammatory in the setting of 3-month post-infection, we didn't see much. We only saw these 3 modules: platelet activation and enriched monocytes in our mate cells, as well as an integrin and cell adhesion molecule downregulated in conventional CDC2s. So overall, this tells us that there seems to be some suppression of myeloid cells at 3 months post-infection in our long COVID patients. But the problem with these modules is we really kind of want to get to the genes and pathways that are involved. So to do this, we performed a totally orthogonal analysis using the BioNet package to build networks of genes that were significantly differentially expressed between our long COVID patients and our recovered patients. And so what I'm showing here is a network of genes that are differentially expressed. Anything in green was significantly downregulated in long COVID. Anything in red was upregulated in the CD14 monocytes, where we saw the most changes. And in fact, if we look, some of these genes were found in those same transcriptome modules, providing an orthogonal validation of this result. But what we also see is networks that are interesting and involved in inflammation. For instance, we see upregulation of IL-1 beta and CXCL8, which are involved in the pro-inflammatory response. So both of these pro-inflammatory pathways are downregulated in monocytes. Related to this, we see ANPEP, which is, interestingly enough, a receptor for one of the less pathogenic coronaviruses that is upregulated. We see downregulation also of RIP2 kinase, RIP kinase 2, which is involved in innate and adaptive signaling and is thought to induce apoptosis. We see downregulation of CD86, which is important for antigen presentation to CD4 T cells, as well as downregulation of genes that are thought to be involved in viral infection, including EBV infection, interestingly enough. So if we really put this all together, what we see is a number of genes that are interconnected, that are involved in innate immune activation and also antigen presentation, that appear to be mildly suppressed at 3 months in those with symptomatic disease 3 months following COVID-19. So now we followed this up with multiple analyses, including some cell-cell communication, but I'm running out of time, so I'm going to skip that slide. And hopefully what I've shown you from our data is that we've generated multi-omic transcriptional and proteomic data sets to investigate the basis of COVID-19. And we found several gene networks and modules that are perturbed in long COVID at 3 months post-infection, which are in agreement with some of the prior published data, but inconsistent with others. And that at the transcriptional level, we're really seeing a downregulation of inflammatory pathways in innate immune cells, particularly CD14 monocytes, and that genes in these pathways encode proteins implicated in human disease, inflammation, and viral infections. But of course, we're still at early stages trying to figure out what this means. So importantly, this is ongoing work that I'm presented here at preliminary stage. We're working to integrate our single-cell RNA-seq, CyTOF, and plasma proteomic data sets. We're continuing to evaluate these communication pathways to see if we can interrupt some of these aberrant pathways that might be inhibiting and inducing disease. And we're assessing the role of adipose tissue as a reservoir in long COVID. So with that, I really want to thank my lab, pictured here. Mine is Sean, who I showed a picture of earlier, and really call out everyone who assisted with this. And this work would not have been possible without assistance from several people in my lab, including Cass Pinedo for running the CyTOF studies. And with that, I will end.
Wonderful. Thank you, Dr. Blish. And next, we'll turn it over to Dr. Rosen.
I'm going to represent the group of PROMISE PI investigators, Dr. Kerwin at Pennington and Dr. Kern at Kentucky, as well as the co-investigators, Jonathan Lee at Harvard and Phil Scher at the University of Texas Southwestern. So I'm going to talk about PROMISE, the pathobiology and recovery of metabolic and immune status. That is our clinical study. But for this talk, what I'd like to do first is give you a brief overview, and Catherine did a nice job summarizing some of the metabolic data that is consistent from published data, at least, that a couple of candidates are altered in metabolism, particularly serotonin and cortisol. And I'm going to talk and focus more on the local effects at the tissue level, and in particular, dyslipidemia. So just to remind you, acute COVID is associated with marked hyperglycemia in a number of individuals, particularly those that are severely affected and are in the ICU. And there's multiple pathogenic mechanisms for the hyperglycemia, but it's clearly worse with obesity. And as you'll hear, that's a common theme during the course of my talk, that obesity not only exacerbates acute COVID, but likely helps to predispose to some of the metabolic changes in chronic PASC. But in addition to, and somewhat underrepresented in the literature, besides the hyperglycemia, is the abnormal lipid profiles. HDL levels are profoundly suppressed in acute COVID, and this is also associated with an increase in triglyceride and an increase in low-density lipoproteins. And this is true also in PASC. So with long COVID, the rate of incident diabetes is increased considerably. This is work from one of my colleagues, Ziad Al-Ali, demonstrating the burden on our healthcare system for individuals who develop long COVID and consistently have not only glucose intolerance, but treatment with anti-hyperglycemics to control their disease. And in addition to the diabetes, though, dyslipidemia again appears and is relatively common. And this is characterized by low HDLs, high LDLs, and an increase in triglycerides. So these are very consistent. In a more recent paper that I didn't show, Ziad also showed that these data sets persisted out to 2 years. So the high triglycerides, the low HDL, and the hyperglycemia are a bad formula combination for the development of oxidized lipids. And we know that the first line of defense for oxidized lipids is HDL. So many of you are familiar with HDL because it's a good lipid carrier in cardiovascular disease. But in addition to that, HDL is a large particle, and it carries a number of other enzymes, which include peroxidase 1 and cholesterol ester transfer protein. And both these are critical in their own ways in making sure that lipids are handled properly and don't end up in tissues where they're not supposed to be. And in particular, peroxidase 1 is critical because it blocks the initial oxidation of lipids to lipid hydroperoxide. And we know from our colleague Grace McComsey at Case that oxidized lipids, particularly oxidized LDL, is higher in those individuals that have symptom complex and so are considered PASC high symptom related. In addition to that, we know that oxidized lipids can generate reactive oxygen species, which are toxic to mitochondria. And in a recent paper, Guarneri has shown that many of the core mitochondrial genes are downregulated during SARS-CoV-2 infection, both in humans and in rodent models. And so the suppression in mitochondrial oxidation is potentially a key factor, not only in acute disease, but in chronic disease as well. And some work from Dr. Kerwin's lab has shown that in those individuals that did not recover from acute COVID, that oxidative phosphorylation of T cells is markedly suppressed compared to those that did recover, suggesting that, in fact, there is this chain of reactions that occurs that might trace back to what's happening with dyslipidemia. So in order to test that, we have a paper, it's on bioarchives and in iScience under revision, in which we worked with the group Inference in Cambridge, Massachusetts, using AI to look backwards and see if we could identify certain predictive factors that led to persistence of symptoms long-term in COVID-infected individuals. And so we did a sort of retrospective case control study in which we found 1,000 individuals from nearly 1 million cases in the Mayo Clinic system that had symptoms at 42 days or later, and matched those to 1,000 individuals that were infected but did not have symptomatology. And there were 3 time points, time 0, which was before infection, and that is the amount that we had in the health records. Those that occurred at time 1, which was during the time of acute infection, and those that occurred at time 2. And we looked at specific simple laboratory measures to assess whether we could predict retrospectively what changed between time 0 and time 1. And this is a very important point. We need longitudinal data, and we need those cohorts that were followed before they got infected to really understand what metabolic changes are occurring. And what we found was that during time 1, at time of infection, triglycerides was increased, as you might predict, and there was a profound suppression in HDL. And it was HDL that was the best predictor of those individuals who went on to develop the long COVID symptoms. So we were particularly interested in pursuing this. One problem with this study was we were only out 6 to 8 weeks. And so we wanted to see more long-term what was going on with HDL. So my colleague Ziad Al-Ali, who's really been a pioneer in the epidemiology of long COVID and its relationship to particular markers, was able to come back and take some of his data sets from the VA system, which he uses, and using BMI matching, he was able to show that HDL not only went down with at 120 days or after the acute infection, but similarly, influenza, post-influenza individuals showed a decline in HDL. And it's been known that even in HIV, HDL levels do decline and may stay down. So this was suggestive that there was high sensitivity, but lack of specificity for a marker for SARS-CoV-2. And then we worked together with a group in Iran at Kerman University, and they had a longitudinal series of individuals at different time points out to 90 days, looking at a number of different biochemical markers, and they categorized them by the difference in their severity of acute illness, moderate, severe, and critical. And you can see on the slide on the left that HDL levels do rebound by 90 days. But interestingly enough, they measured peroxidase activity in the serum and found that that rebound did not occur as consistently as HDL did. So one thought is that HDL is just a surrogate marker of peroxidase activity, and that the persistence of high oxidized LDLs may be related to a persistent decline in the ability of the enzyme to prevent lipid peroxidation. We investigated this a little further in the Recover cohort in Maine. We have 120 subjects, and we looked at HDL levels as predictors of symptom complex using the JAMA symptom complex from the Recover cohort. And interestingly enough, baseline HDL at the time they entered Recover in Maine was not predictive of subsequent symptom complex magnitude or frequency, but the HDL levels at the time of their most recent response to their questionnaires was negatively related to symptoms. So the lower the HDL, the greater the symptom complex. And we could not find additional parameters from the lipid studies that we get in Recover that actually could identify those individuals. So with that in mind, as part one, just to remind you, insulin resistance, type 2 diabetes, and incident diabetes are more common in PASC and may reflect a number of different components, possibly including adipose tissue dysfunction. Suppressed HDL may be a marker of some lipid toxicity and probably has high sensitivity but low specificity. Whether it's a surrogate marker for peroxidase activity needs to be examined. And whether HDL may reflect chronic metabolic inflammation still needs to be determined. So with that in mind, let me move to the second part of the talk, which is our investigation into the mechanisms that underlie metabolic dysfunction in post-acute sequelae of COVID. And our hypothesis has been that PASC participants with a high symptom complex are characterized by insulin resistance, adipose tissue inflammation, high endotrophin levels, which I'll talk about, T-cell bioenergetic fatigue based on the work of Dr. Kerwin, and that possibly this is related to viral persistence. And I should point out that we regularly meet with Catherine and Charles Roberts, and we're all sort of in the same group trying to identify whether virus persists in adipose tissue. And as Catherine mentioned, we're still in the early stages. Okay, so Our design was a case-controlled design of 60 participants from 3 rural states in the Recover program. So we belong to the I-SCORE network, which is 11 individual sites in a hub network, which is run out of West Virginia University, Sally Hodder. And we proposed to get 20 Recover participants from each of 3 sites, Maine, Kentucky, and Louisiana. And the idea was, one, rural individuals are generally underrepresented, so these were 3 rural states. And 2, we had a mix of ethnic and racial individuals that would be optimal for trying to understand the differences in PASC and recovery versus non-recovery. So it was a case-controlled design, and it was based on a 2-to-1 ratio of 40 cases that had symptom scores of greater than 12. So we developed a symptom score in collaboration with the DRC at Recover that was similar, although not absolutely identical, to what was published in JAMA, but reflected very much the same 8 most frequent symptoms that Catherine also mentioned. And our primary design was actually to look at T-cell mitochondrial function using oxidative phosphorylation on the seahorse as a major outcome measure. But secondary outcome measures included T-cell markers, glucose tolerance, DEXA, body composition, as well as the adipokines and cytokines, and viral studies, including nasal swabs, blood, and adipose tissue, as well as some tertiary outcomes, which included spike protein analysis from David Wald, adipose tissue secretome histology, and RNA-scope, and endotrophin. So where are we? Currently, we've had 17 fully consented. Almost all have been at the main site. We got started early, and early being in July. So as Catherine said, we still have a ways to go in terms of getting full data sets. But they're matched by age, sex, and body mass index through the data center at the Pennington Bio Research Center. 7 OGTs have been completed, 6 adipose tissue biopsies, and body composition by DEXA. And these are the pending studies. And we'll talk a little bit about endotrophin and its role, as well as the studies on viral persistence. So I'm going to show this data. It's very preliminary, but I want to give some idea of where we're going, because a couple of these studies are unique to the pathobiology program, and they're also excellent for hypothesis testing and further follow-up. So this is the first 8 subjects who had DEXA body composition studies, and although not statistically significant, there's a trend towards higher body fat in those that had the higher symptom complex. And in addition, percent visceral fat was also trending in the higher range in those that had PASC positive symptoms. Now, this is interesting because the BMI are matched almost identical, and yet we see these differences in visceral and total body fat distribution. And so this is an important area that we need further investigation. It comes back, though, to the issue of whether obesity itself predisposes individuals to long COVID and more symptomatology. And so in a way, it's sort of reverse causation. Is the obesity actually causing the symptom complex, or is this symptom complex from viral persistence driving more metabolic fate? And that really needs to be determined by larger numbers of individuals. Similarly, the oral glucose tolerance test was performed on 8 individuals. And again, those that had slightly greater visceral fat and more symptomatology had a higher area under the curve mean glucose. So very consistent. What was a little surprising to us, and it's picked up because of the way we do DEXA body composition studies, bone density was lower, both total body area BMD, spine bone density and femoral hip bone density were all lower in the past positive individuals. And this was consistent with some biochemical studies we did on their plasma, which showed that urinary CTX, which is a marker of bone resorption, was increased. We see in chronic inflammatory states some reduction in bone mass. We also see reduction in bone that can occur from large amounts of visceral fat with secretion of cytokines that are detrimental to the skeleton. So this is an area that hasn't been further investigated, but important for us to try to understand whether long-term complications, including osteoporosis, may occur. And then blindly evaluated were initial measurements of surface markers, including CD26 and CD3 and CD8, which suggested that these individuals do have some inflammatory component. And again, this is very preliminary and may or may not be consistent with some of the literature. So one of the things we've done in our study is do wedge biopsies of fat tissue. And we've been so impressed, and I have to have a shout out to Recover participants. They've been fantastic. They want to participate in this study, and they're willing to go ahead and do these, including individuals that are fully recovered from long COVID. So on the far left is the biopsy done by a dermatologist. Here is the 2 grams of fat tissue that we get. And then Sam Costa, which is a very talented PhD student in my lab, has been extracting fat tissue, culturing them, and then looking at the results. And Charlie Roberts has helped us in addition by doing RNA scope. This is just the first subject that we sent him fat tissue on to look for a virus, and unable to find it. But obviously this is very early. This is a single participant. One of the interesting things is that these individuals, when their fat biopsies were analyzed by targeted QRT-PCR, so we were very interested in what this fat tissue was doing at 48 hours in culture. And what we found was an upregulation in those that had symptom complex of some of the classic pro-inflammatory markers, TNF, IL-6, and CRP. In addition, collagen 6A3. And I want to just briefly divert to mention collagen 6A3. So collagen 6A3 is induced by hypoxia and HIF-1 alpha, and it's a major extracellular matrix that is secreted from cells. Once it's secreted, it's broken down by proteases to form the compound called endotrophin. And endotrophin is a major inflammatory marker that can actually persist around fat cells, and particularly in fat cells that are inflamed from obesity. And you can measure endotrophin in the plasma. So endotrophin's major activity is the recruitment of macrophages. Macrophages can also make endotrophin, but endotrophin levels have been reported to be high in end-stage renal disease and in type 2 diabetes in particular. And so we wanted to know whether or not this was the case. It's also present in lung tissue from individuals with pulmonary fibrosis and those with chronic pulmonary disease from COVID. So we set out to see, using different cohorts, whether endotrophin could predict markers of severity of symptoms. And again, using a similar symptom complex, we were not able to show any relationship between endotrophin and the severity of post-acute sequelae symptoms. We were able to see in those that had lung disease and we either had respiratory arrest or those that had low flow oxygen, there was an increase in endotrophin, which was significant. So
excuse me, Dr. Rosen, we're a bit over time. Are you able to finish up in about 2 minutes?
Yeah, I can. So, and endotrophin levels, we were unable to identify a change in individuals in the UAB cohort we used. And I just want to show and finish with this slide, which shows some of the immunohistochemistry of endotrophin around fat cells, right around the margin of fat cells. And so we're pursuing that now. And I want to finish with a slide that just suggests that SARS-CoV-2 infection can indeed result in hyperglycemia, increased oxidized lipids, and this could be a vicious cycle that enhances metabolic dysfunction. So our future directions are we're going to 7 bridges to test the predictive value of HDL. We'll expand our participant number, and we'll examine our secretome analysis. And this is the group of individuals that we've been working with. So thank you very much. Sorry to be a little over.
Wonderful. Thank you, Dr. Rosen. And with that, I'd like to hand it off to Dr. Nath.
So I'm going to talk about clinical phenotypes of long COVID, pathophysiological mechanisms, and opportunities for intervention in 20 minutes or less. Okay. So I have no disclosures. That's one thing that happens when you work for the government. You usually have no disclosures. Sometimes I wish I actually had some. Okay, so the first thing is we don't even know what long COVID is. So there are a lot of definitions of long COVID. There are 3. The term long COVID was actually coined by patients. And so what they found was it was pretty simple. You know, they got COVID and then they weren't getting any better. They said, well, I think I got long COVID. And interestingly, that's the term that is stuck. And NIH came around and came up with this term of post-acute sequelae of COVID-19. And people do use the term PASC, but really it's a mouthful. And then the WHO came up with post-COVID-19. So the definition that WHO has is the one that most people are still using, and that is the continuation or development of new symptoms 3 months after the initial SARS-CoV-2 infection, with these symptoms lasting for at least 2 months with no other explanation. But in reality, none of these definitions are good, because what you have is you have 2 buckets of individuals. You have the first set of individuals who developed COVID, they were admitted in the hospital, and they got all kinds of end organ damage. They come out and they're not healthy. It takes them a while to recover. And sometimes they don't recover completely, but it's not because there's something ongoing that's new, but rather it's the end organ damage that occurred during the acute illness. Then you have a second set of individuals. And these are the individuals that I think we need to worry more about. And these are individuals who had relatively mild infection. They never got admitted to the hospital. They recovered or almost recovered. And then they say a few days, weeks later, they develop these new symptoms and they relate it to COVID. Okay. So the pathophysiology of that has to be really different than the first group. Okay, how many people develop this thing? So this is the CDC data. And what they claim is that if you just look at the entire US population, about 6% of the US population has long COVID. That's huge. If you look at those individuals who had COVID and now complain of long COVID, they claim there's about 11% of individuals and currently. So that's still a huge population. And it affects those in the age group where you're most productive of your life. So the socioeconomic consequences are huge. If you look at the symptoms that people have over a period of time, you'll see that the neurological symptoms seem to persist. Because that's the lower box over here. They seem to persist, and 6 months to a year later. A lot of these symptoms of anosmia, respiratory stuff, this all disappears over a period of time. So the neurological consequences are really a major problem with this disease. Okay, so if you look at the second group of individuals, and I don't know, we need to come up with a better term for them, but over here I just use long COVID for them. You can divide them into 4 buckets. Okay. And so one is that have predominantly exercise intolerance. They can take a flight of stairs and then get totally fatigued. Yeah, those that have cognitive problems, they may not even be aware of it till they actually go back to work or a challenge. And then they realize, oops, I'm not thinking the way I used to. or they can develop an episode of major depression, sleep disorders. And there's another group that develops dysautonomia or POTS. And that's a separate category by themselves. And then there are individuals who have pain syndromes. But there's overlap between all of them. So you can have predominantly one or the other, but you can have multiple symptoms. So we wanted to study the neurological manifestation. So what we did is we looked at a cohort of 173 patients, and we have all kinds of criteria, and we ultimately studied 12, brought them over here. What we found was that 100% of them had cognitive difficulties and fatigue, and then they had a variety of other neurological symptoms. Okay, what about age? So here, this is an interesting study. What they did was they looked at individuals who develop any kind of respiratory infection and those who develop COVID. So the blue line is COVID and the red is other respiratory infections. At every age group, you can see that the chances of developing COVID is much greater than with any respiratory infection, except when you turn 70 years of age. Okay? But that age group is toast. Doesn't matter what infection you get, the news is bad. Okay, now these guys did some PET scanning and they said, okay, let's look at the brain and see what happens in these individuals. So on the left-hand side, this is a composite scan of normal individuals. You can see with the fluorodeoxyglucose, all these bright colors means there's increased uptake. It's a nice uptake here. You look at the brain from a COVID patients and you can see there's decreased uptake in the front and in the basal ganglia. The posterior parts of the brain are relatively spared. And another study looked at it a little bit more closely, and they claim that if you look at the bottom of the brain, actually there is more dysfunction than other parts of it. So what are the possibilities? The possibilities are one, that you reactivated some kind of virus that was there. The second is that maybe you have a persistent viral infection, you now got rid of it. Or thirdly, maybe there's immune dysregulation, and that's driving the pathology. Or there could be interrelationship between all three of them. So here's evidence of persistent activation, and that is a reactivation of Epstein-Barr virus. There's a study from Yale, and they looked at patients with long COVID, and you can see that the antibody titers to Epstein-Barr virus are greatest in these long COVID patients. And they looked at two different antigens here. Okay, what about persistent infection? So we thought, that the infection is in the nose, it infects the nasal mucosa, so it'll just go up the cribriform plate and it'll infect the brain. That's what our original thinking was. It turns out it doesn't occur that way. In fact, if you look at the nasal mucosa, there's a lot of virus. In fact, you can see it here in this electron micrograph. There are these black dots here. Each one of them is a viral particle. But if you go into the olfactory bulb or tubercle, you don't find virus at all. Very rarely do you find some individuals who have a little bit of virus. And that's because the virus infects the sustencular cells and not the olfactory nerve. If it infected the olfactory nerve, it would go in, no problem. But if it infects these support cells, then it sticks around, it'll cause edema, and that's why you get the loss of smell. We looked for virus in the brain at autopsy, we didn't find it. We looked very hard with all kinds of different techniques. We couldn't really find anything. There are others who have reported that they can find virus, but when they do find it in small quantities, or they find it very, very rare. So this is a beautiful paper from some of my colleagues here at NIH, did a Herculean task. And what they found was that, yes, you can find a lot of virus in the respiratory tract less than 14 days. But even if you go there greater than a month, you can't find anything. If you look at the brain here, less than 14 days, only 32 copies compared to what they found there. And then if you go 31 days or later, can't find anything. So I don't think the virus itself is driving the neurological symptoms. There's got to be more to it. Now, this is a interesting case report of two children who they claim had transplacental spread of the virus. And you can see this massive atrophy of the brain here. And they found viral antigen here in the brain at autopsy. The immunostained it here, that's one. And nucleocapsid. And this is a study by Dr. Egan, who is the National Institute of Aging. And what she did was she looked at the papilla of the tongue, and she claims that she also found some evidence of viral antigen in some of these patients. And they were And this is like 40 weeks out from the original infection. So some of the loss of taste could be related to viral infection. And this is another group from Boston, and they find that they can find some spike protein in the serum of patients with COVID, suggesting that there is persistent antigen. But just the presence of antigen alone doesn't necessarily mean that that's the cause of the symptoms. A number of viruses, you can have persistent antigen. They may actually do nothing. But the fact that you can find something, I think one needs to explore what the consequences might be. So what about immune dysregulation? Okay, so I'm going to show you data on both antibodies and macrophages. So this is a group, they looked at patients who had depression and cognitive problems, and they used a ligand called TSPO, which binds to activated microglia in the brain. And they find that those individuals who have these cognitive problems have increased uptake, suggesting there's more microglial cell activation. And at autopsy, we looked at these brains and we found the same thing. There is a lot of microglial cell activation, both in the gray matter and white matter. And this is a group from the Netherlands, again, showing great activation of the brain using this a ligand that is very similar to TSPO. It binds to activated microglia. We looked at brains of individuals who had died of COVID. They had died acutely. However, we got them from the New York Medical Examiner's Office. And these were individuals who died early in the pandemic, either at home or in a subway. They were just found dead. So they never went to the hospital, because their respiratory symptoms were very, very mild, if any at all. So had they not died, their symptoms would have persisted. They would have developed symptoms of long COVID, I believe. So anyhow, we looked at their brains because the brains were highly abnormal. And you can see this blood vessel here, and this is a high resolution MRI scan, a 7 Tesla scanner. You can see thickening of the blood vessel, a small blood clot over here, and then another cross section, you can see a little microhemorrhage here. So 3 different types of pathology in the same blood vessel. This is at 11 Tesla scanner. Now you can see the pons and you can see the olfactory bulb, and we immunostained it for fibrinogen, which is a very large protein and should never enter into the brain. So all this brown staining is fibrinogen. You can see that around the blood vessel, there's a huge amounts of staining and then kind of peters out. Even in the olfactory bulb, there's loss of fibrinogen. So they're leaky blood vessels. And I think that's a major problem. We looked at substantia nigra, and you can see here on high resolution MRI scan, you can see loss of the signal here in the substantia nigra. And there is fibrinogen leakage in the substantia nigra as well. Histologically, we found a lot of macrophages in the perivascular region. That's no surprise, because if you have all these proteins leaking from the blood, you need the macrophages to remove them. But once the macrophages get in there, they're bad actors. They don't leave. And these activated macrophages can cause a lot of damage. Found activated astrocytes. What we did not find is T cells. Okay, very few T cells around the blood vessel, none in the parenchyma. If this was a viral encephalitis, you would find a lot of T cells. There are no T cells. But we found a lot of activated platelets here sticking to the blood vessels. Some of them are clogging the blood vessels there. And when we quantified all the histopathology, we find that most of it is at least there's more in the posterior parts of the brain and the hind parts of the brain as compared to the other parts of the brain. We looked at the blood vessels more closely, and what we found was there's activation of adhesion molecules. You can see that over here. We found deposition of complement, and we found deposition of some IgG, but predominantly IgM. So we think that this is an antibody-mediated damage to the endothelial cells, and that's what's making it leaky. But why would the antibodies attack the endothelial cells? Okay, now going back to the same cohort of patients that I told you earlier we brought to NIH, 100% of them, when we looked at their CSF, they had activated B cells in their CSF, and they antibody-secreting B cells here. And there were other kinds of immune abnormalities as well in these patients. Now, this is a group from Hopkins, and they looked at antibodies to ACE2. Okay? And what they found was that in these COVID patients, you could find IgM antibodies to ACE2 out several days. I mean, here 3 weeks, and but they don't switch. You should get a class switching from IgM to IgG. That didn't occur. So the IgM kind of persists, and that it correlates with the findings I just showed you. But why ACE2 is the question. So one possibility is that what these patients are producing are anti-idiotypic antibodies. So you have the antibody against spike protein. If you produce an antibody against that antibody, is going to mimic the spike protein and bind to ACE2 receptor. But if you look at the vasculature in the brain, this is, you can see the blood vessels everywhere. Right? So if you damage the blood vessels, you can attack almost any part of the brain and any cell you can get at. So we looked at the neurons in the brain. You can see here in the cerebellum, there's loss of striae, and that's because there's loss of Purkinje cells. And so there is neuronal damage that is occurring because of this. We were very interested in looking at the brainstem. And particularly, I was interested in the pre-Bartzinger complex, because remember, these patients had died in their sleep, and their hearts, there was no pathology there. So the question is, could they have died from a central cause? And the Bartzinger nucleus complex controls respiration. And we found that there was atrophic neurons, activated microglia, and there's a phenomenon called neurophagia, and that's what's killing these neurons. So I suspect that they died from central hypoventilation, which is also called Ondine's curse. Okay. It's a nice fairy tale that I think you should look up. I'm not going to tell you the entire story. Okay. Now, there are some interesting other abnormalities that people have described. This is an interesting paper. And what they claim is that it all has to do with tryptophan uptake. And the tryptophan uptake from the gut is impaired, so you're not getting serotonin, and that's the reason for it. I mean, it's an interesting observation. They've done a lot of work, but I doubt that one single metabolite is going to explain the whole complicated disease syndrome. And patients who are on SSRI, a lot of these patients are on SSRIs, it doesn't really do much to them. Now, this is a paper from Yale that just got published, and they claim that cortisol levels are decreased in patients with COVID. Okay. And it's the same one that also showed EBV activation, antibody activation. Okay, what about acceleration of neurodegenerative disease? So the UK Biobank looked at these individuals, and they showed that there is atrophy of the brain in those individuals who get COVID compared to the counterparts who did not develop COVID. And if you look at blood markers, you will see that neurofilament levels are increased and amyloid levels are decreased, which coincides as biomarkers for Alzheimer's disease. And some patients develop POTS or dysautonomia. So when they stand up, their blood pressure can go down and heart rate can go up. And there are multiple other types of dysautonomias that can occur in this population. And some can develop a small fiber neuropathy as well. Yeah. And then there are a variety of pain syndromes, headache being a common one, quite common. And the causes are sometimes difficult to figure out. So the potential for loss of symptomatic treatment that one can do, so you can intervene, there are a number of things you can do to help patients. And certainly there are a lot of different immune modulatory drugs that can potentially be used depending on what you find in these patients that is dysregulated. And we have an ongoing, I'll show you. Another thing you can do is prevent long COVID. So vaccinations do help. So if you get one vaccine, and in this meta-analysis, you can see going to the left doesn't do much. You get 2 dosages, you start going to the left. 3, it really favors it. And I have had 5. All right, so what we're doing here is
I'm sorry, Dr. Nath, if you're able to wrap up in a minute or so, that would be great, so we can get on to the Q&A. Oh, wonderful, thank you.
So, and I'll conclude by saying that direct invasion of the brain by SARS-CoV-2 is rare and may not explain the neurological complications. Neuroimmune dysfunction is driven by activation of innate immunity, immune exhaustion, and antibody-mediated phenomena. And clinical trials with immune therapy should be considered in these patients. These are all my acknowledgements. I'll stop here.
Thank you, Dr. Blish, Dr. Rosen, and Dr. Nath for these wonderful descriptions of the ongoing work, the ways in which Recover is highlighting this it is clear from your discussions that one of the challenges we face is that both the definition of long COVID itself, as well as components of long COVID, like brain fog, are largely patient-driven, as is appropriate in many ways, but something that where we are describing it as we go along, trying to understand it. It is clear that there are responses to this infection in the form of activation of T cells, changes in antibody responses, changes in hormonal responses, as well as metabolomic changes that might, that appear to play a role in, or at least are associated, whether they are causal or not is not clear, but they are associated with the presence of symptomatic long COVID. There have been a number of very interesting questions, and one of them from a sufferer is, what is the average duration of these symptoms, and is there, for people who have persistent symptoms as long as a year, does then the subsequent expected duration, what would that be? And I know this is an area where the pandemic itself is only about 3 years old now, so we may not have much by way of answers, but I would just like to pose it to each of you as to what has been, what in your judgment is the course of the long COVID and resolution of the biology, the pathobiology, as well as the symptoms. Catherine, do you want to, Dr. Blish, do you want to take a?
Sure, Catherine's fine. I mean, that's, I think, kind of intrinsically an unanswerable question, because I don't, I mean, my hope is that this is what Recover as a consortium is going to be able to do. You know, in trying to come up with, you know, a consistent even definition or a consistent description or a consistent biomarker. I mean, it's been very, very difficult. And I think many of us believe that is because there is heterogeneity, like long COVID isn't one thing. And I mean, both Drs. Rosen and Nath alluded to this, like Dr. Nath had his 4 kinds. You know, maybe there are 4 kinds, maybe there are 8 kinds, maybe there are 10 kinds. And I think that what is likely is that the underlying pathophysiology driving each of those is going to be slightly different. And the resolution therein is going to be different. I mean, in our data, we do note that, you know, altered taste and smell and cough actually resolve almost universally. But brain fog seemed to actually, you know, be getting worse. This is, so, you know, if you have lost your smell, hooray. If you have brain fog, we don't know what the natural history of that is going to be. And that's where we're going to need these numbers and these people looking across different cohorts with different BMIs, different ethnicity, different other risk factors to try to figure out, you know, are there unifying groups that we can find where we can say, you know, really the era of personalized medicine for this subset that might help resolution and for this subset. But I just don't think that we have a good enough grasp, or at least I don't feel like I have a good enough grasp to say like respiratory COVID is caused by X and it's going to get better in 3 months. I mean, I would give my eye teeth to find that, but like at least in our data and in my reanalysis of other people's data, I can't find a unified definition.
Just one follow-up. There have been a number of questions asking about, say, pediatric age groups, young adult age groups. Dr. Nath showed that, for instance, if you're over 70, perhaps the risk is greater. Have you seen differences in these immune responses across the lifespan? Would we design different kinds of trials for different age groups or age cohorts of people with long COVID?
I mean, I think that's a great question. And I think there was even one in the chat about differences in sex. And the problem with that is our cohort is 100 people, and and we don't have every analysis on every person. And by the time we get into our subgroups, we're not seeing many differences by sex or by age. But, the finer we dice that, the more power we lose to find significant differences. So we haven't seen a dramatic difference in our cohort, but we haven't looked that hard because we're underpowered.
Thank you. Dr. Rosen, I think one of the questions that you alluded to as well is that some of the people with hormonal metabolomic changes like low HDL, insulin resistance, type 2 diabetes are at a higher risk anyway of COVID and of long COVID. And so the question was whether this is pre-existing, whether this may be unmasking syndromes in people with a propensity, or whether this was driven by a reaction to either a persistent viral infection, a reservoir like fat. We know it's not persistent in the brain. So the Lancet article with Ali in the VA seemed to suggest that if you have persons, that cases and controls pre-infection seem to be fairly similar. So I just welcome your thoughts on how much you think this is a reaction to the virus, which is resulting in some metabolomic changes, whether it's direct or the immune-mediated changes. And I know you talked about this, but there have been questions in the chat about it as well.
Yeah, so that's a really difficult question, and we don't know the answer. Certainly, when you compare those non-infected versus those that are infected, the increase in incident diabetes in long COVID patients is higher. The question really becomes whether or not that is exacerbated by body or host metabolism, that is obesity, which predisposes individuals to a more chronic inflammatory type of adipose tissue, which could then lead to either viral persistence or more chronic inflammatory markers. And we don't know yet because we need longitudinal data. We need individuals who, and this is where Recover comes in so much because it not only tells us more about the long-term duration of the disease in answer to your first question, but it also has a sub-cohort of individuals who were not infected and then got infected during the last couple of epochs. And those individuals are probably the most valuable group of individuals because they're not infected, they're being followed, they get infected, and then they have post-COVID symptomatology. I do want to emphasize that some people do get better, and it's really important, although we don't know why they get better, if this is the natural history of long COVID, or if they're taking agents offline through the internet, through any friends, colleagues, that may be ameliorating their symptomatology. A lot of my people are starting to take metformin based on the randomized controlled trial. Do these interventions or vitamin D or any of these that NO1s are being tested by individual patients, whether that helps, we don't know, and we need a registry to really help us understand what those agents are and if they're any better. Recover is the first step, and it really is extremely valuable because I think we're still early and we still need that natural history. And finally, it's heterogeneous. So everybody's going to be a little different. And the host response is going to be very, very important to this virus.
And the longitudinal study of all of these changes as well. I think there was one question about if I had a bad long COVID response, but I've recovered and I'm infected again, am I more likely to have long COVID? And I don't know if you have an answer for that.
Yeah, well, there's multiple infections lead to a greater risk of long COVID, but whether you're fully recovered and then you get another infection, whether that predisposes, I don't think anybody knows.
Dr. Nath, you had clearly discussed many immune-related ways in which the changes in the brain might be explained. Do you think the hormonal and metabolomic changes that Dr. Rosen was speaking about might have a role as well? Is there a way? Many of the autopsy information we have right now is clearly from more of an acute or subacute setting. So how relevant may these different streams be to the brain fog of long COVID?
So I'm pretty sure hormonal and metabolic abnormalities will be found in this patient population. And there's some work, Dr. Iwasaki from Yale presented at the Keystone meeting, and some of it is included in our paper that just came out in Nature, but not all of it. And there she did show both types of abnormalities. She showed that cortisol levels were decreased, and if you look at women, their testosterone levels were decreased. And she claiming that some of those things probably make a difference. And then, another parallel is ME/CFS, this chronic fatigue syndrome, where people have looked at these kinds of things. Overlap. There again, you can find all kinds of metabolic abnormalities there too. If you look at do metabolomics of the CSF or blood. So I think there are a lot of parallels there, and you'll find all these things. You look for them, you'll find them.
There have been a lot of questions about, subjectivity versus objectivity and whether there could be serological biomarkers. You had shown some data on NFL, and we know that NFL crosses the blood-brain barrier and circulating levels do reflect what's in the CSF. What are your thoughts on that as a marker of acute injury? Yes, we have this information. What about persistent neurological impacts from post-COVID?
Yeah, so this is an area that's very near and dear to you, and and you know that better than anybody. That now you have NFL, a number of things you can measure, and there are a lot of literature now on these things. They're not showing that in progressive neurological disease, no matter what the cause is, if there's ongoing neuronal damage, neurofilament levels, light, heavy chain, and GFAP, these things are becoming really good biomarkers. They don't quite tell you what the underlying disease is, but at least tells you there's progressive disease. So, and now this assays become so sensitive that you can measure them in serum. You don't need spinal taps on all of them. So I think you're absolutely right. These patients should be monitored, and the presence of that indicates ongoing pathology.
There's another question. I'll let any of you answer that, about studies underway looking at persistent or other decline mitochondrial function as an explanation. We could also decide that some of these are for a subsequent R3 seminar, because these are whole areas that we could explore. But in case any of you wanted to take that.
Well, there was that paper,
Yes.
that I illustrated that suggested that mitochondrial genes were downregulated, and I think there's a bit of evidence that was shown at Keystone from a group in the Netherlands that showed decreased mitochondrial function in muscle tissue from muscle biopsies of long COVID people. But
so it's a tricky thing to study, right? Because you've got if you get muscle deconditioning, you're going to get, you're not moving around, you're not doing anything, the muscle is going to show decreased metabolic function in the mitochondrial related thing, but that just could be all deconditioning. So one has to be careful with how to interpret mitochondria. We did a lot of mitochondrial DNA sequencing, gene sequencing out of the muscle in chronic fatigue patients. But if you do the seahorse experiment, you'll find abnormalities. If you look at it, it's all deconditioning.
Thank you. Those are clearly questions still to be answered. There was a lot of interest also on ongoing treatment trial information and what the panel might think are the most promising interventions to prioritize in studies, focusing here, I guess, on brain, immune, and metabolic dysfunction. There were, for instance, suggestions about transcranial direct current stimulation for brain fog, about LDN and Maravoric. So I just wondered if one of you wanted to address. I know, as far as I know, these are not ongoing trials. I didn't find anything on clinicaltrials.gov, but if perhaps the panel is aware of efforts to set up something or what they would think would be most promising interventions. Yes, go ahead.
Yeah, I was just going to say the metformin study is really interesting. The previously published paper, and a number of my people have been put on metformin now as one way. It may actually improve mitochondrial function as well as all the other things that metformin does. And I'll only say that providers are desperate for interventions. So it's really important that these clinical trials be done and done well.
What about immune?
I'm sorry to interrupt. We just have about 2 minutes left. Would it be okay if I shared the closing remarks? Great. So thank you so much to all of our presenters today, and thank you for our audience for attending this seminar and for engaging with the Q&A. We recognize we didn't have time specifically for some of those questions. So as a reminder, both a recording of today's seminar will be available on recovercovid.org within a few weeks. And we'll also be posting a Q&A document that has responses to the questions that we received today, including the ones we didn't have time to address. Okay, we have some upcoming seminars, although I believe we don't yet have a defined date, but we're working on some additional ones for the future, and we hope you return. Finally, you'll see a short survey come up on your screen asking for your feedback on this seminar. We'd appreciate it if you could take a minute just to fill out this brief survey. With that, thank you all and have a great day.